Hi, this is David Friedman and my co-host Dave Burkus for TCA Crossroads, where entrepreneurs and investors connect. And I'm real excited today to see you, Dave. How are you today?
I'm doing fine, Dave. This is a great day. It turns out to be a sunny day as well. It was 90 degrees out today, not knowing when your listener is going to listen to this. And I know we both have some news and we have some great guests. So why don't you first start with the news?
Well, the news is I just was able to work on a new strategic affiliate relationship with Frontier Angels. They're going to be coming on as a fifth strategic affiliate. Graham Conran is the chairman of that group. I'm real excited. I've known Graham for years through the Angel Capital Association, and really solid, good players, smart. So I'm excited. I'm really excited about that one. And you have good news too. You have a new book, right?
Just published my 15th book, The Leader's Toolkit, available on Amazon and anywhere else. It actually prints in color when you order it on Amazon, which is a pleasant shock for me.
How cool. Well, everybody should avail themselves of the opportunity to hear Dave directly through these podcasts, his social media, and read the book. You'll get these pearls of wisdom that will help you grow into that billion-dollar company. And if you're an investor, make sure you don't make bad decisions. How's
Nice.
that, Greg?
Okay, let's bring on our guests and let's see what we can do.
All right, I'm going to bring them on. I have now brought on two guests. This is the great dyad of George Carpenter, who's now president of TCA Orange County, and also the deal lead for Holoclara. And we have Andrea Choe, who is the founder and CEO of Holoclara. And I am really excited because we're going to have this very interesting discussion that I believe will help both the entrepreneurs understand what it takes to be successful in getting funding, and hear from George on what the angel investors or early stage investors can do to make sure they find winners such as Holoclara. So if you'd like to introduce yourself, Andrea, you can have your 15 seconds of fame. George, if you want to say something, and then I'll turn it over to Dave for the first toss-up question.
Well, thank you so much for having me. My name is Andrea, as you just introduced me, and I'm the CEO and co-founder of a company called Holoclara. And what we do is we are leveraging a multi-million year evolutionary relationship between long-lived symbionts and humans to find the next great blockbuster drug.
Those are worms, right?
Correct.
Okay, I just want to make sure for all the people who have to now go on that thesaurus and find out what the heck we're talking about.
Let's first talk to George.
So I'm a serial entrepreneur. I've had 4 companies, all of which I kind of stumbled into, and over $1 billion in exits. And I grew up in Iowa, and we have a farm. So I've had some of my best friends are worms.
Hey, nice. You have them by name?
No.
No, okay.
They're living inside of him as we speak.
Exactly.
Oh.
Exactly.
Feeding thoughts to him. Yeah.
That's the punchline.
Well, let's start by asking George how he found Andrea and how this was organized as far as the investors. That's a good thing for the investor side.
Well, this would be like 2021, 22. And so we were all in our igloos, getting on the internet. But there was already kind of a buzz around Andrea from entrepreneurial circles, both in LA and Orange County. And one of the things that we identified early on with Andrea is she has that absolutely high conviction that an entrepreneur needs to have to go through all the crap that you go through in starting a company. And she seemed, whether well-advised or not, committed to doing this. And so we just kind of followed her lead. It was an easy deal to put together. We had lots of syndicate members. And I think over time since then, Andrea, tell me if I'm wrong, but I've sort of been able to stay in touch with her. And since in all 4 of my companies, I've made all the mistakes that are possible, my role is basically to just see, what she's doing and make sure she doesn't make all my mistakes.
Nice.
But she has not.
And one more question to you, George, before we get to Andrea. So I understand that you led the deal with Orange County. How did you combine the various chapters to be able to make the ultimate investment for TCA?
We had, like I said, there's already kind of a substrate of interest among many people, and word had gotten out on this deal. So literally, it was just a process of engaging with LA and Pasadena and other folks. I think we even had some folks outside of TCA, but it was an attractive deal, and it was early on. So one thing I'm hoping we'll hear is kind of what's happened since then with Andrea, because she's made some stunning scientific progress.
Ooh, David?
Yeah, so I'm interested. I'm going to go to Andrea. So I'm going to advocate for you because I remember seeing you back in 2021, early on, talking about worms, and I thought that was really interesting. And you've come such a long way. But how did you pick George, or did you pick TCA as an investor, or was it just he was around and available? I'm trying to understand your thinking about choosing the right investing group, because what I understand is we, TCA, collectively have invested $750,000 in you.
But the answer that I always have is we find each other, right? So I think that's how George and I found each other as we were having conversations. TCA, the idea of an angel community, has always been interesting to me, because I feel like everybody should be able to get into these deals. And I love the community that these various TCA chapters have built. And the fact that there's a podcast, right? The fact that you guys get together and host events, it's wonderful. And then also being squarely in LA, which, as you know, a lot of biotech is not. So being kind of central between Caltech and being able to pull in Amgen folks, I think that LA is a really great growing space for biotech. So I think just naturally being in the same place and supporting each other is what brought us to each other. And then George obviously has a worm that is telling him to bridge the conversations to happen, right?
You mentioned Caltech. Is any of this intellectual property licensed from any of the universities?
Yes. So Caltech is where I did my doctoral work that led to this discovery. So this is licensed from Caltech, and then we've had incredible support there, not just in terms of licensing, but we continue to go back there. We have more service agreements there than with any other biotech startup. And whenever I get someone new to set up an agreement, they always say the same thing. We're like, we took it to the head of development, and they made a comment. Was it you again? So, there is a lot of wonderful resources there that we're leveraging, from cryo-EM to their drug development center. We have incredible chemists there. We actually did manufacturing there, right? So it's just a wonderful place.
I learned a new term from Andrea last week at her Harvard TED Talk: worm influencer.
Yes.
I do have a question to you, Andrea, again. When you look at TCA, you look at George or the other investors, talk about the biggest challenges you had prior to funding. And then somewhere during this discussion, I want to talk about what you have accomplished after funding. But what were the early challenges you had in working with a group like TCA? I don't want to put George on the spot, or you, but in working with George.
Your question is, what are the challenges we've had at the company before funding, or you're saying specifically?
During the funding journey, during the funding journey, when you're working with TCA, what were some of the challenges you had before you actually got the funding? Were there any challenges? Was it really easy? Were there issues? I'm just trying to understand that.
No, it was very sweet and straightforward. And actually, when you were asking us about this, I couldn't even recall the details of it, George, because it just happened so naturally.
Nice.
So I'm trying to come up with a more substance for your answer, but I can't, because it just kind of came together.
Let me go back then to the post-funding, but I'm curious about a couple things. Your Harvard talk, TED or TEDx?
TEDx.
Yes, okay. So let me ask that question then about post-funding. And I've already heard that you were really enthusiastic about what you were doing, which is amazing. And now the question is coachability. And so let me ask if you're using any of the members of TCA or others as an advisory board, and if so, kind of tell us how that works.
Well, I have George, right? We're always in conversation, we're always in dialogue. And to be honest, I don't know if you just have one of those Jedi type coaching mechanisms where it's never clear that it's being coached. It's just natural dialogue about what are the ebbs and flows of the company, what are the strategies, how are we prioritizing, what's next for us. So I think all of those just naturally come up in conversation.
Is it a formal advisory board or just George?
Just George.
Oh,
One thing I've said though, and Andrea's heard this, but like of all the CEOs that I've worked with, she's one of the best at recruiting and working with and really working hard, top-notch lions of medicine, science advisors, people that can actually help her move this thing forward at the molecular level. So she is for a first-time CEO, I'm really impressed with how much she works her advisors and gets the most out of them, and that's part of where some of the scientific progress has come from.
So what I'm also hearing when I listen to both of you, and it's like you both like each other, you seem to trust each other. So the ability to work together and be on that same page is really critical. Were there any disagreements you had during the funding process or even now in terms of strategy, in terms of tactics, in terms of financing? Or was it just easy? I'm trying to help the listening audience figure out how to best deal with an investor, and also the investors, and how to best deal with the entrepreneur. And sometimes there's an abrasion. Did you have any abrasion? Is it disagreements? How did you handle them?
I hate to be boring, but I've learned never disagree with a worm influencer.
Well, I also come fully loaded. So as George said, my advisors, I have on the regulatory side, former chief counsel to the FDA on my board, who's happy to join a call and talk about his role and his guidance. I have Carl Labelle, who is former chief of staff to Roger Perlmutter, right? So, I have a lot of great clinical guidance. I have a head of R&D that's a rock star in the LA ecosystem, who's been chief CEO before, chief scientific officer, chief development officer, has really demonstrated his worth at Amgen for many years. So I bring a lot of people to the table, I think. And I think from there, I'm able to share in conversation with George how we're thinking about this. And I don't think you've ever countered any of their guidance either.
I think my point is, when it comes to the science, I'm a dog watching TV.
Well, that's fair if you know where your role is. So that means you have a formal board and an advisor, but not an advisory board, just informal.
Oh, I have many advisors, yes, including Carl Labelle, who I just mentioned, is one of them. Stan Lapidus, who's just extremely well known for his role in, Exact. Also, if you've ever taken Cologuard, which is the colonoscopy screening measure, he's titan in that field. So we also have KOLs that are the top KOLs in the world for multiple different allergic conditions. So I think that I have incredible advisors that help us along the way.
Okay, so you have not organized them as a single board, you just use them one by one as you need them.
Yes, I guess you could say that we have our scientific advisory board, right? But we also have our clinical advisory board. And then the clinical advisory board is really comprised of the KOLs and also people that have clinical operational experience.
How are you playing with the FDA? Are you going to have to get de novo clearance when you finally have the product out, or is this 510K?
So right now we're not engaging with the FDA because we were capital efficient, went to Australia for our phase 1 clinical trial. We are actually contemplating how to get the fastest proof of concept, which may again be somewhere that's not in the US. Although our KOL for eosinophilic esophagitis is in the US, and he wants to be the PI on trial. We've submitted an IIT. He tends to score well, being the top KOL in the world, but also Australia has the highest incidence of allergies in the world. So I think that there is a strong rationale for going back there to be able to get the fastest proof of concept.
What are your milestones over the next 12 months? Let's talk about where you are now and where you want to go. And what do people have to look forward to in terms of seeing a commercial product eventually?
Yes. So to go back to a question that maybe we didn't answer yet is post-funding, what did we do? And so from that point forward, we really started doing the pre-phase 1 preclinical work. There were a couple of major milestones from that point of having preclinical data, knowing what our molecule looked like, what its structure was, whatnot. But part of that was one, scaling it. No one had taken such a unique compound before and had ever scaled it. And so this was really a hero's journey that involved Brian Stoltz, who's a brilliant synthetic chemist at Caltech. You know, his claim to fame has always been when someone says something's impossible to make, he finds a way to make it. Not to say that that was our issue, but we were able to reduce the number of steps that we needed to synthesize with his help. And also being able to find an incredible CRO to make this within our time and budget. So that was number one, with the purity that we needed. And that was a huge unknown. It worked out. It worked out incredibly. Then from there is, can we dissolve it? What is it like? It's an oral drug. What does it taste like? So, you know, it is flavorless and it also dissolves incredibly well. So we almost had the opposite issue where a lot of drugs where you sit there and you try to spin it for a long time, it's not dissolving, which was my experience in academia with other compounds that I've been studying. You're sitting there with a vortex all day long, just listening to your podcast or playlist. But this one actually was so dissolvable that we transported it in a special container so that moisture wouldn't get in. From there, the pharmacy had a really easy time being able to dissolve it and give it to participants. But the step one was actually just making the compound. Then step two was all of our IND enabling preclinical tox, which is multiple species. And, you know, with up to 14 days of dosing, we saw no signs of tox at 10,000 times the therapeutic dose.
Wow.
So then you design a clinical trial. We talked to trial sites all around the world. The question was, are we going to do an IND? We talked to US trial sites, we talked to Australia and everywhere in between. Really liked Australia because they had a lot more experience. Also because of the 43% tax back, the tax reimbursement, right? So it was just incredible to work with them. And what we did was we designed a SAD-MAD trial, 56 participants. And so that really was a lot of last year. So what we've done was that we gave it to participants, tracked them for safety. And because this is the first in man for this molecule, you have to stage it where there's a sentinel being dosed first. Then you have to track and then recruit. So you have to really be able to synchronize the timing of your trials and reading out the labs and calling your clinician who's on the other side of the world to sit there and do the readout with them to make sure that you can now advance, and keeping that all on time for something that's a first in class. It just was great that we were able to do it within the budget and the time frame that we had. So then there's more. As they say, wait, there's more. The next biggest challenge was figuring out the human molecular target. The number one challenge for natural isolates is, what is the active molecule, which sometimes people never figure out. And the other part of it is, what is the target? How do you know that it's only in mice? What if you have a great therapy for mice and it doesn't translate to human? And how can we go back and make sure your tox studies are relevant? Do those species that you did your tox studies in, what if the target doesn't exist in them, right? So this is the $1 million question, is how do you do that? And traditionally, with drug development, it's really been challenging to do them the traditional way. And George was saying, I have talked to a lot of Nobel laureates about Holoclara, one of them being David Baltimore. Caltech, I brought this to him. He was like, you're not going to find that person here, Andrea, because we don't ever have to think that way. We have the luxury of not having to deal with those sorts of things here. So you're going to have to go somewhere else. I went to other places, and our molecule is somewhere between the size of caffeine and acetylcholine. It's really tiny. So if you probe it, how do you know you're not blocking the active site? You probably are with something really, really small. So then the challenge is, how do you figure out where it binds? Yeah. And we use this technology from the Macmillan lab, who is this brilliant scientist originally out of Caltech that went to Princeton, where you can tag your small molecule with an iridium cage, and then you put it in cell types, which we want to start with human cell types. Then you flood it with a second compound, diazurine, and then you flash with light, and things that are only within 10 nanometer scale, diazurine will label and grab everything around it.
That was exciting, because I have been involved with trials before, and I have been involved with the FDA. And so to me, I was following you, even though I didn't know the names of the compounds. I knew the process that you have to go through. You're nowhere near done, but it certainly shows that you're using the advisors, you're doing the work, you're doing it in Australia, you're getting a tax credit or a credit, all the right things.
So and then we're using the newest technology that came out only 2 years ago to find our human molecular target. Yeah. And then also show that it was in the species that we did our tox studies in.
You mean you couldn't have done this before 2 years ago?
I don't think so.
Oh, wow.
That's part of the drama of this scientific breakthrough that Andrea is talking about. I mean, she's talking about atomic level imaging and being able to discover not only how this acts in the body, but maybe even some theories, Andrea, about how the messaging works.
But I do have a question that I want each of you to address. If you can think about one thing, this is for our audience, both the investor as well as the entrepreneur. If you can think of one thing that people should do in the startup ecosystem, what could they understand, or what should they understand when looking for funding or when investing in a deal? So I'll start with you, Andrea, and then we'll go to George, and then Dave will wrap. So what should they understand in trying to fund, relative to your journey in funding?
Is this something new? Is this something completely new? Is it a novel target? Is it something that pharma is desperate for?
Cool. Okay, so that becomes transformational. I totally agree. Flipping to George now. George, what would be what your understanding? What should you have investors think about as they look
I'll pick up right where Andrea left off. One of the things she did right off the bat was she said, let's focus on these multi-billion-dollar drug franchises that are trying to do immunomodulation with like a Dupixent. And if we can replicate their transit of the arc, but with a better molecule, and we think there is one in worms, that's not only a potential blockbuster category killer, it's also a potential platform for many other drugs. So I think of this like Eli Lilly had a GLP-1 project that they canceled, and now they've had to spend billions getting back in the GLP-1 game. I feel like, Andrea's basically building that, I wish I had a worm kind of molecule. And that, obviously is great for investors because it means a lot of people will want to do strategic partnerships.
Transformational and could be a platform. You're hitting all the right things, George. Nice to see.
Any final words, Dave?
No, I think we've asked a good number of the things, and it's obvious that Andrea has many more things to tell us. It's too bad we have to make this a collapsed broadcast of only a limited period of time. It was really a lot of fun listening to what you had to say, Andrea, and thanks, George.
And for the viewing audience, just remember that what we're seeing between Andrea and George is a collaboration. and the ability to collaborate in a non-confrontational mode and to have that same focus on the end result gives you a really good result. So understand that it's not one against the other, but this is, use your investors, use your advisors, this to the entrepreneur, wisely to be able to help you build your company. And the investor, find a good entrepreneur that's willing to be coached, listen, and willing to use you for a sounding board. So,
Nicely said, David. Now, clock us out.
all right. So till next time, have a good one. Thank you for joining us, Andrea and George. Appreciate that.
Thanks all.